This video covers the chemical reactions of cellular respiration and fermentation. And we can summarize these reactions as being reactions for ATP synthesis. And synthesis means to make. And so these are the chemical reactions that make ATP. We've established before that ATP is the energy molecule of the cell. And we'll talk a little bit more about how it's made and how it's the energy molecule of the cell a little bit later on. But first we need to take a look at what this word energy means. The kind of generic definition of energy is just the ability to do work. And work usually means the ability to move something. It's not always movement, but it's the ability to make something happen. And oftentimes we're going to be talking about something moving that requires energy to happen. So let's address why our cells need energy. So why do our cells need this ATP molecule? Well, we've already talked about a couple of reasons why cells need ATP. For example, we talked about active transport in the cell membrane PowerPoint. Remember, active transport is the primary active transport. And our vesicular transport, which includes endocytosis and exocytosis. We also need we also need ATP for things like muscle contractions. And muscle contractions include the muscles involved in breathing, the muscles involved in moving blood around your body. So your cardiac muscle, your heart muscle needs ATP in order to beat to move blood around your body. It also has to do with other skeletal muscles. So moving your body around. Skeletal muscles are also the ones involved in breathing, et cetera, et cetera. Just any of the movement you do is going to require ATP. We also need ATP for things like nerve impulses, which really rely on these active transport processes here. Now, we could go on and on. There are a lot of reasons why cells need ATP. Oh, one more I want to mention are things like anabolic reactions. So remember, anabolic reactions are ones where we take some smaller molecules and we bond them together to make bigger molecules. And so, like I said, these are just some of the examples of reasons why cells need energy in the form of this ATP molecule. Now, there are essentially two main types of energy. There's kinetic energy, which is the energy associated with motion. All right, so think about a ball rolling down a hill has kinetic energy. If you swing your fist to punch somebody, your arm has kinetic energy. A bowling ball rolling down an alley to knock down pins has kinetic energy. All right, so it's just the energy associated with something in motion. Potential energy is also kind of known as stored energy. So this potential energy, we can also kind of call it stored energy, meaning that it's not necessarily performing work at this time, but something could change and make it perform work. So for example, if we look at this example down at the bottom here, we see this person with a ball at the top of the hill. This ball has potential energy, so it's not moving. It doesn't have kinetic energy yet, but if this person lets go or even pushes this ball down the hill, as it starts to roll, it now has kinetic energy. And it's important to note that these two forms of energy can go back and forth. So energy can be converted from potential energy to kinetic energy and from kinetic energy to potential energy. We mentioned how there are basically two main types of energy. So we have kinetic and we have potential energy. Well, there are some subtypes of kinetic energy and some subtypes of potential energy. And the ones that we're going to focus on in this lesson, the subtype of kinetic energy we're going to focus on is something called mechanical energy. Other forms of kinetic energy can include things like light and heat and radiation. Potential energy, we're going to focus on the potential energy stored in chemical bonds. So we're going to refer to chemical energy as the specific type of potential energy that is related to this concept of ATP and making it happen. All right, so mechanical energy definition is very similar to kinetic energy. So it's just the energy involved in that movement of an object. All right, so we can look at kind of two different examples I have here. So we have driving a car and a muscle contraction. And I'll relate these together in a moment here. So chemical energy, remember we said, is a form of potential energy. So it's stored energy. Remember, stored means potential energy. And so chemical bonds actually are able to store energy in them. If you break those bonds, you typically release that energy to be used by something else. If you make bonds, you're actually storing that energy to be used at a later time. So let's go back to our example of driving a car. All right, so when you drive a car, your car in motion has mechanical energy. All right, the car itself has kinetic energy. There has to be mechanical energy to move the wheels to make the car go forward. But that energy has to come from somewhere. And if you've ever run out of gas, you know what happens when you don't have energy going into your engine in your car to make it move. And so for your car, the chemical energy is actually stored in molecules of gasoline, right? So gasoline is just an organic compound. And it's made up of a whole bunch of carbons bonded together with hydrogens. So gasoline is essentially just a really big hydrocarbon. And each one of these chemical bonds is storing energy in it. So here, stored chemical energy. And so what you do is you take this chemical and you put it in your car, and then your car has different components that are able to break those bonds. And remember, when you break bonds, you release energy. And that released energy gets transferred to the axle and the wheels of the car in order to make it move, which is how you actually move your car and get that kinetic energy. Now, our body does something very similar, but instead of gasoline, because if we drink gasoline, it will kill us, but instead we can eat different kinds of organic compounds, okay? So here I've got the example of sugar, but we can really get the stored energy from lipids, proteins, and other carbohydrates as well. All right, so just the stuff that we eat that gives us energy. Once again, these are just organic compounds, so very similar to gasoline, but different. So don't go drink gasoline. But these are organic compounds, and our cells have specific enzymes that allow us to break down these organic compounds. And as we break down the organic compounds, we're breaking those chemical bonds and releasing that energy. And so in our example here, that released energy gets transferred to our muscle cells, allowing them to contract, which gives us the ability to move our muscles. So we can, flex our biceps, or we can change the shape of our chest cavity so that we can breathe, or our heart can contract in order to move blood around our body. As mentioned previously, ATP is the energy molecule of the cell. So you can think of ATP and energy as being essentially the same thing. And ATP stands for adenosine triphosphate. So that's where the letters ATP comes from. And ATP is a molecule that is able to store energy in its chemical bonds. And remember that these chemical bonds are potential energy. So if we look at our ATP molecule down here in this picture, so this is our ATP molecule. We can also kind of draw it, I kind of draw it like this just to make it a little easier for me to visualize what this looks like. So I just have my adenosine as the A, and then I've got my 3 phosphate groups kind of bonded from that adenosine. All right, so essentially the adenosine and ribose, this part makes the adenosine part. And then tri means 3. So 3 phosphates. We can see there's phosphate number 1, phosphate number 2, and phosphate number 3. Now, all of these bonds have chemical energy stored in them, but the bond between the second and third phosphates is an especially high energy bond. All right, so it's this bond that really stores a lot of energy. And so this bond is going to be made and broken as we basically make ATP and then use ATP. So ATP can actually be made from a molecule called ADP. Which stands for adenosine diphosphate. Where di means 2, and you'll notice that there's 2 phosphates, plus a phosphate just kind of hanging out on its own. And when the bond is formed between the second and third phosphates, we can form our ATP, which remember is our adenosine triphosphate. Where tri means 3, and we've got 1, 2, 3 phosphates. Now, this reaction is reversible, so we can also draw a backward arrow, because remember, we can essentially make and break that, that bond between the second and third phosphates in order to either store energy or release energy. So in order to make that bond, there has to be an input of energy because you have to store that energy. Okay, so we can think about energy. In this case, it's going to be energy from the organic compounds that you eat. So remember, this is things like lipids, which are the fats, proteins, and carbohydrates. And so what cells do is they break the bonds between the atoms in these molecules, and they release that energy. And what happens is that energy goes into this reaction, and then that energy gets stored in this bond between the second and third phosphates. Now, in the opposite reaction, where ATP gets broken down into ADP and an extra phosphate, that energy can then be released. And that released energy can go to something like a pump. So for primary active transport, we have this transport protein called a pump that moves things like ions across the cell membrane. Or maybe it goes to a muscle cell in order to make it contract. Or maybe it goes into an anabolic reaction in order to form a bond between two other molecules that are getting, put together to make a bigger molecule. So, in essence, we can kind of think of ATP as like a rechargeable battery. ATP itself is like a charged battery. All right, so this is a battery that is ready to go. You can put it into something and make something happen, whereas ADP and phosphate would be like a dead battery, where you've used up all the charge in the battery, but it could be recharged and used again as long as you get an input of energy. You can think about this with something like your cell phone, for example. Your cell phone, you can't just plug it into a wall and use it. There's actually a battery that's an intermediate between the electricity and the wall socket and the actual applications on your phone. So in order to charge your phone, you have to have an input from the electricity from the outlet. That outlet goes in and it charges the battery, and then you can use the battery to play Candy Crush or make a phone call or surf the internet or something like that. But once again, you've got this battery as the intermediate, and that's exactly what happens in our cells, where ATP is kind of this intermediate between these organic compounds and the cells themselves. Because we can't just take sugar and rub it on a cell and make the cell have energy and do something. It has to be first converted to ATP to then be used by the cell in order to fulfill a function. So we've mentioned how we can get energy from these organic compounds. So we can get it from our fats, which is our lipids. We can get it from our carbs, which are our sugars. And we can get energy from our proteins as well. And if you're not familiar, calorie is actually a unit of energy. So this is like the units, like if you were to say centimeters or pounds or grams, it's just similar to that. So calorie is just a measure of how much energy is in something. And so we get calories from fats, carbohydrates, and proteins. But notice there's no ATP, right? So if there's no ATP in the nutrition facts, how do our cells get ATP? How do we get ATP into our body? And the answer to that question, how do we get this ATP, is by doing these chemical reactions known kind of collectively as cellular respiration, which converts the carbohydrates and the fats and the lipids into ATP. To understand energy, we have to understand a little bit of physics as well. And so thermodynamics is just basically the study of energy and how energy behaves. And so the first law of thermodynamics, which you've probably heard of before, is that energy cannot be created or destroyed. You may have also heard this referred to as energy being conserved. This law is also called the law of conservation of energy. And what this means is that there is a finite or set amount of energy that exists in the universe. And there's no input of energy, so there's no extra energy coming into the universe. But we're also not losing energy as well. So we're not creating it, we're not destroying it, but we can transfer the energy from one molecule to another and from one form to another. And this is what happens during those chemical reactions of cellular respiration, where we're not creating energy and we're not destroying energy. We're just transferring that energy from the organic compounds to ATP. And then from ATP to whatever the cell needs that to happen. So it could be, it could be transferred to a pump or a muscle or something else. You've probably heard of something called a food chain or a food web. And this picture here is showing us a food web. So this is showing us a group of organisms and showing how basically they eat each other. And when you eat something, you're basically taking its biomass. And what that means is you're taking its molecules, so you're taking its atoms, but you're also taking its energy. And so when we eat something, our food gives us carbon and hydrogen and oxygen, but it also gives us energy. And so these food webs and these food chains are just showing this energy getting passed from one organism to another in the form of kind of juggling these different molecules and breaking bonds and making bonds. So let's jump into these different types of cellular respiration. And things get a little bit confusing here because cellular respiration technically means kind of these two different forms of respiration. So it technically means both aerobic and anaerobic respiration. It gets confusing because sometimes aerobic respiration is also just called cellular respiration, but cellular respiration does technically refer to both the aerobic and anaerobic pathways. So for the sake of this course, I'm going to refer to cellular respiration as this general idea of ATP synthesis. And then when I'm talking about the two main pathways, I'm going to use the terms aerobic and anaerobic respiration in order to keep those two pathways separate. But just know if you look in like a textbook or something, sometimes when it says cellular respiration, it's also referring to specifically aerobic respiration. And I don't know why they do that. It's confusing. But I didn't write all the textbooks, unfortunately. So if we look at these kind of these two chemical pathways over here, so the key difference is aerobic and anaerobic. And so we need to look at what these terms mean. So aerobic versus anaerobic. Aerobic just means that oxygen is required in order for it to happen. And we can abbreviate oxygen as O2 because the oxygen gas that we breathe in is just made up of two oxygen atoms bonded together. Anaerobic, when we look at kind of Latin roots, the letters AN mean without. And so anaerobic is just the opposite of aerobic, in which oxygen is not required for it to occur. So we can go back over here and say that aerobic respiration is, we can say with O2 or no O2. All right, so that's the key difference between these two types. It's just whether oxygen is present or not. But we're also going to learn that there are other kind of specific differences. So for example, aerobic respiration occurs in kind of four main steps. The first step is glycolysis. The second step is the oxidation of pyruvate. The third step is the citric acid cycle. And the fourth step is known as oxidative phosphorylation. And oxidative phosphorylation has kind of like two sub events happening. The first one is the electron transport chain. And the second one is known as chemiosmosis. Now, anaerobic respiration has a different set of chemical reactions, kind of. The first one is actually still, and I'm going to change colors here real quick. So the first one is actually still glycolysis. So glycolysis is actually step one of both pathways. But for anaerobic respiration, the second pathway or set of reactions is known as fermentation. And then that's it for anaerobic respiration. And we're going to go over all of these in more detail on the following slides. Before we get into the specifics of the reactions of aerobic and anaerobic respiration, we first have to look at something called redox reactions. And redox reactions are actually two types of reactions that typically happen together. So a redox reaction refers to a reduction reaction and an oxidation reaction. So the red stands for reduction and the ox stands for oxidation. So in a reduction reaction, a molecule is going to gain electrons. And those electrons have to come from somewhere. And so those electrons come from other molecules. So remember, electrons are one of those subatomic particles. So atoms have protons, neutrons, and electrons. And remember that electrons are involved in chemical bonding. And so atoms can give up electrons. They can also accept electrons, and that kind of changes their their bonding properties to a degree. So in an oxidation reaction, a molecule loses an electron. And then in the corresponding reduction reaction, a molecule is going to pick up or get those electrons. So we're basically just transferring electrons from one molecule to another molecule. And we can see that in this picture over here, where we have a molecule that is being oxidized. So an oxidation reaction, this molecule is losing this electron, and it's losing this electron and giving it to another molecule. Since this molecule is gaining that electron, it is going through a reduction reaction where it picks up that electron here. Now, it's a little bit hard to remember what these different terms mean. So I've got this mnemonic that I have used since high school, and I have a master's degree in biology, and I still have to use this mnemonic to remind myself what happens during a reduction reaction and during an oxidation reaction. So the mnemonic is just the words oil and rig. And so oil stands for oxidation is loss or losing electrons. And the rig stands for reduction is a gain of electrons. And as I mentioned, these two reactions typically happen together. So we just kind of abbreviate it and call it a redox reaction, where one molecule gains an electron from a different molecule that loses an electron. And we're going to be talking a little bit more about these redox reactions, specifically when we talk about these molecules that are used in cellular respiration reactions called, we're going to be talking about NADH and FADH2. And these molecules are actually called electron carriers. So we're going to talk about how electrons get transferred from molecules like the organic molecules in your food to these other molecules, and then those electrons get carried to something called the electron transport chain, which allows us to make ATP. So now we're moving in and focusing on our aerobic respiration steps. So we're going to start by kind of introducing this aerobic respiration, and then we'll break it down into the different categories of reactions. So then we'll talk about glycolysis, oxidation of pyruvate, et cetera, et cetera. So first, let's look at the overall equation. So I'm actually going to draw a couple of arrows here, because one arrow doesn't really do this justice. There are actually dozens of reactions that happen between the left side of the arrow and the right side of the arrow. But this is the overall reaction in the sense of what has to go into the reactions in order to make ATP, and then what do we get out of them. All right, so we're going to start by looking at our C6H12O6. So this is our glucose, which is a sugar. And we use glucose as kind of like a model molecule, because glucose molecules go through every single step of aerobic respiration. But later on, we're going to talk about how glucose, remember, is not the only molecule that we can get energy from. So really, we're using glucose as just kind of an example, but really this could be any sort of organic nutrient. All right, so this could refer to other carbs, so other sugars, things like starch, fructose, sucrose. It could refer to our lipids, so our fats and oils, or it could refer to our proteins. So we can actually take all of these and put them through these reactions of cellular respiration. It's just that other carbs, lipids, and proteins don't necessarily go through every single step of cellular respiration. They just go through some of them. So we'll talk about that a little bit more later. The other thing we need are some oxygen molecules. So O2 just stands for oxygen. And then the outputs or the products are going to be our carbon dioxide. So our CO2 is carbon dioxide. H2O is water. And then our energy is going to be our ATP molecules. All right, but as I mentioned, this isn't just one reaction that happens. There are dozens of reactions that happen between the left side of the arrow and the right side of the arrow. And as I mentioned on a previous slide, there are 4 main steps to cellular respiration. So glycolysis happens first, followed by the oxidation of pyruvate, which is also called the transition reaction, followed by the citric acid cycle, which back in my day was actually called the Krebs cycle. But they've updated the name because the new name actually makes a lot more sense because it tells you a little bit about what's happening in that cycle because there is citric acid involved. And then the 4th step is going to be our oxidative phosphorylation, which has these kind of 2 sub-steps to them of the electron transport chain and chemiosmosis. When you learned about cell organelles, you probably learned that the mitochondria is the organelle or structure needed for ATP synthesis. And that is absolutely true if we're talking about aerobic respiration. So this kind of diagram here, it's showing you the cytosol, which remember can also be called the cytoplasm. And then we have our mitochondrion here. And so we're going to be talking about these kind of different locations. So first of all, that glycolysis reaction is happening in the cytoplasm. The other reactions that we talked about, so the oxidation of pyruvate, the citric acid cycle, which I guess I should label that here, and then the oxidative phosphorylation is all happening in the mitochondrion. So let's take a little bit of a closer look at the structure of a mitochondrion. So I'm going to draw just a big circle for our cell. All right, so we'll say that this is the cell membrane here. And I'm not going to draw this to scale because the mitochondria are obviously very small, but I'm going to draw it kind of big just so we can label the different parts of it. So this big kind of oval inside is going to be our mitochondrion. And we can kind of draw a little squiggly line inside. All right, so all in all, this whole thing here is the mitochondrion. And mitochondrion is just singular for mitochondrion. Plural would be mitochondria. So that's the difference there. So out here, kind of between the outside of the mitochondria, but inside the cell membrane, we have our cytosol, which remember is also called the cytoplasm. Or we can call it the intracellular fluid. So it's just the goo inside the cell. And our glycolysis reactions are happening out here. Now, the mitochondrion itself has a couple of different regions. So it has an outer membrane, it has an inner membrane, and this inner membrane you'll notice is folded. And so the folds are actually known as cristae. And the reason why it's folded is it helps to increase surface area. This is something you see a lot in biology. So more surface area usually means more room for chemical reactions. And so this is especially important because this is where the electron transport chain is located. So we can actually kind of draw the electron transport chain is basically made up of these kind of transmembrane proteins. So these integral proteins that allow different substances to kind of cross over that membrane. And so the more surface area we have, the more room we have for these electron transport chains. All right, so this is the electron transport chain. Now, inside the inner membrane, we have something called the matrix. All right, so the matrix is kind of the gooey filled center of the mitochondria. And then the area between the inner and outer membranes are called the intermembrane space. So what happens is glycolysis happens out in the cytosol, like we mentioned before already. And then the products of glycolysis actually go into the matrix. And so inside the matrix, we have our second step, which is the oxidation of pyruvate. And the third step, which is the citric acid cycle. And then remember, step 4 is the oxidative phosphorylation, which includes the electron transport chain and chemiosmosis. And so for our fourth one, that oxidative phosphorylation, what happens is you actually have materials that are going from the matrix, so we'll draw it over here, from the matrix out to the intermembrane space, but then other stuff going from the intermembrane space back into the matrix. And so oxidative phosphorylation actually happens kind of across the cristae of that intermembrane space, or I'm sorry, of the intermembrane of the mitochondria. So let's start by taking a look at our first kind of set of reactions known as glycolysis. Now, glycolysis itself is actually an anaerobic process. So remember, anaerobic means without oxygen. So we do not need oxygen to occur. Now, not to confuse you, because we're talking about aerobic respiration here. So we're talking about making ATP in the presence of oxygen. So why are we talking about this process that is anaerobic and does not require oxygen? So remember when we talked about these reactions of cellular respiration, we talked about how there are two pathways. So we have the aerobic pathway and we have the anaerobic pathway. And remember, the aerobic pathway we said requires oxygen, and the anaerobic pathway does not require oxygen. And then we listed the kind of the main steps of these pathways. And the first pathway, or the first set of reactions in both of these, was glycolysis. So glycolysis itself is actually an anaerobic pathway. But it's the start of both the aerobic and anaerobic reactions. What happens after that is where things actually start to differ. Now, glycolysis occurs in the cytoplasm, so we do not need a mitochondria in order for glycolysis to occur. So mitochondrion is not needed in order for glycolysis to occur. We don't need the mitochondria until we get to these later steps of aerobic respiration. Now, if we look at the word glycolysis, we can actually break it down into glyco and lysis. And glyco is the kind of like a prefix having to do with sugars. So think of things like glycogen, glycocalyx. These are all things that are related to sugars. And then we know from learning about hydrolysis reactions that lysis means to break something. So the term glycolysis actually means to break sugar. All right, so it means to break sugar. And that's exactly what happens during this chemical reaction. We're going to start by looking at our glucose, which is our C6H12O6, which is a sugar. And we are going to break that sugar down into something else to get these reactions started. So we're going to start by looking at the reactions of glycolysis. Now, as you can see from the image, there are several kind of like intermediate reactions that happen between the beginning and the end of glycolysis. And this image doesn't even show all of them. There are, I think, about a dozen reactions that happen between glucose going into the reactions and pyruvate coming out. So we're going to look at kind of the big picture. So what is the overall input or reactants? And then what is the overall output from these glycolysis reactions? So as we go through glycolysis and the oxidation of pyruvate and the citric acid cycle and oxidative phosphorylation, we're going to be tracking kind of three main components. So the first main component is going to be the carbon-based compounds. And so I'm going to use the color black because carbon is found in coal. And so whenever I think of carbon, I think of black. So we're going to start by looking at a 6-carbon molecule, starting with glucose. So remember that glucose, the chemical formula is C6H12O6. We're going to primarily focus on the carbon. So we're just going to say that this is a 6-carbon molecule. Now, the end result is that 6-carbon molecule gets cut in half, and we end up with 2 3-carbon molecules called pyruvate. You might also see this called pyruvic acid. That's the same name. It's just 2 different names for the same molecule. But notice how we start out with 6, we end up with 2 3-carbon pyruvate molecules. So I know it's going to be kind of hard to see on this dark background. So let me switch over to the white pen real quick. So glucose is our 6-carbon compound, and we end up with 2 3-carbon pyruvates. Additionally, during these reactions, we have some of those redox reactions happening. And so what happens is as glucose gets split up into pyruvate, some electrons get transferred from the carbon molecules to this molecule known as NAD+, and it converts it to something called NADH. And so as you can see over in the diagram here, what happens is that electrons get picked up during these redox reactions. So I actually like to draw some little dots next to the NADH to remind myself that there are electrons that have been picked up by this molecule. In addition to these electron carriers, so NADH is an electron carrier, remember? So we'll talk about why that is important when we talk about the later stages of aerobic respiration. But in addition to these electron carriers, we are also looking at our ATP, ADP kind of reversible reaction happening. Now, in glycolysis, we actually have to have a little bit of an input in energy in order to get the reaction started. So we actually have to put 2 ATP into the reaction to start breaking that glucose. And so we can think of this as kind of like a -2 for ATP because you have to invest some ATP to get things started. But in the end, we end up making 4 ATP. So we can think of this as a +4. So even though we have to invest or put in 2 ATP, we ultimately get 4 ATP out. As we mentioned on the previous slide, we have to put 2 ATP into the process to get glycolysis started, but we ultimately get 4 ATP out. So if we look at net, so net ATP production, net means what is the profit? So how much did we get after we account for the fact that we had to use 2 ATP to start the process? So we make 4 ATP, but we had to use 2 in order to get it started. So ultimately, the process of glycolysis produces 2 ATP molecules. And this process is called substrate level phosphorylation. And this is different from the oxidative phosphorylation that we're going to talk about a little bit later, which requires the electron transport chain. All right, so this is not oxidative phosphorylation. This is called substrate level phosphorylation. So we talked about how during glycolysis we made some NADH. And so NAD plus was reduced to form NADH. And remember, we drew those two little dots on NADH to remind us that it had picked up some electrons along the way. So did NAD plus gain or lose electrons? And we can go back and remember our mnemonic, and we can say that NAD plus was, remember, oxidation is loss, reduction is gain. So NAD plus was reduced, meaning that it gained electrons to form NADH. And I'll add those two little electron dots on there. So if we look at the products or the outcomes or the outputs of glycolysis, what happens to those? So why do we care about the ADP, the NADH, and the pyruvate? So first of all, ATP, remember, is just energy. So we've already made some energy that the cell can use. Now we could stop there, but we could get a lot more ATP out of that glucose molecule if we kept going. And we'll talk about how that happens on the following slides. The NADH, remember, is an electron transporter, and it's got those electrons, and it's going to transport those electrons to the electron transport chain. So we're going to talk about this NADH more in a little while. So right now, just kind of keep it in the back of your head that this NADH is hanging out, holding on to some electrons for us. And then last but not least, we've got our pyruvate, which is our 3-carbon compound. And remember, we actually have 2 of these because remember we had a 6-carbon glucose that got broken down into 2 3-carbon pyruvates. Now, this carbon compound of pyruvate, we can still get some more energy out of it. And so that pyruvate is going to keep going through the additional stages of aerobic respiration in order to allow us to make some more ATP. And remember that glycolysis actually occurs for both aerobic and anaerobic respiration. So the fate of glucose depends on whether there is oxygen present or whether there isn't. And so we're going to talk about what that pyruvate does if we have oxygen or if we don't. Now, you're probably familiar with the fact that humans have to breathe. If we stop breathing, we die. But have you ever stopped to think about why that is the case? Why do we have to breathe? We know that we have to inhale to take in oxygen. But why do we need oxygen? Well, we need oxygen in order to make ATP using that aerobic pathway. All right, so we have to breathe in to get oxygen into our body because we need that oxygen to go through the oxidation of pyruvate, the citric acid cycle, and oxidative phosphorylation. Without those, the pyruvate would have to go through the anaerobic stages, and we'll find out at the end that the anaerobic pathway is just not as efficient as the aerobic pathway. So that oxygen allows us to get a much greater return on investment with regards to how much ATP we can make per molecule of glucose than the anaerobic pathway. Right? So we need that oxygen, and more specifically, we need that oxygen to make ATP. Now, exhaling is also really important because remember that we looked at the overall equation of cellular respiration, and we said that you had to have some sort of organic molecule. In our case, we're talking about glucose, plus some oxygen in order for that to occur. Now, the end products of cellular respiration include carbon dioxide, water, and energy in the form of ATP. So the main reason why we exhale is actually to get rid of this carbon dioxide. So some carbon dioxide dissolved in your body fluids is actually a good thing. You need some in there, but your cells are constantly making more of it. And if your carbon dioxide level gets too high, it can actually cause problems, including something called acidosis. And so you'll learn about that more when you get into acid-base balance in anatomy and physiology type classes. Most people use the terms breathing and respiration interchangeably, as if they mean the same thing. But in reality, they actually have different definitions to them. So breathing refers to basically moving air in and out of your body and gas exchange between things like your lungs and your blood vessels and between blood vessels and the cells. So breathing is really just moving air between different areas kind of in your body and exchanging that air with the external environment. Respiration refers to the cells actually using that oxygen to extract energy from those organic molecules. And I say oxygen, but we can make ATP, we can do anaerobic respiration without oxygen. So respiration really just means that cells are extracting energy from organic molecules, whether oxygen is involved or not. So remember that breathing is just moving respiratory gases like oxygen and carbon dioxide in and out of your body, whereas cellular respiration is actually taking those organic compounds and extracting energy from them to make ATP. All right, so we've talked about how during glycolysis we make this molecule known as pyruvate. So now let's assume that we have oxygen. So what's going to happen to this pyruvate? Remember, pyruvate is this 3-carbon compound. So it's got 3 carbons in it. Well, pyruvate is going to go through the remaining steps of cellular respiration. So it's going to go through the oxidation of pyruvate, which is also called the transition reaction, the citric acid cycle, which is also called the Krebs cycle, although that term is starting to become kind of older and not being used anymore. And then last but not least, it's going to go through oxidative phosphorylation, which involves the electron transport chain followed by chemiosmosis. Now, in addition to needing oxygen, these steps also require mitochondria in order to occur. So we're going to be talking about the specific areas of the mitochondria where these different reactions occur and why that's important. All right, so after pyruvate is made during glycolysis, it moves into the matrix, which is that kind of gooey filled center of the mitochondrion, in order to go through the remaining steps of aerobic respiration. Now, for each glucose molecule that goes into glycolysis, we end up having 2 pyruvates, because remember, glucose is a 6-carbon compound that gets broken up into 2 3-carbon pyruvates. So we've got 2 pyruvate molecules that are going to go through the oxidation of pyruvate. So looking at the overall reactants and products of the oxidation of pyruvate reactions, first let's take a look at our carbon compound pyruvate. So remember, pyruvate is a 3-carbon compound, and it starts out by combining with something called coenzyme A, which we abbreviate as CoA. And what happens is it ends up making a 2-carbon compound called acetyl-CoA, or acetyl coenzyme A, plus carbon dioxide, and carbon dioxide, of course, has 1 carbon in it. So we've taken 3 carbons in pyruvate, and we've broken one of those carbons off into carbon dioxide, and then we've made this 2-carbon molecule known as acetyl-CoA. Additionally, we've made some additional NADH. So NAD plus picks up some electrons to make NADH. And once again, I'm going to draw those little dots there to remind myself that the NADH is carrying some electrons. So the NAD plus was reduced in order to form NADH. So NAD plus gained, remember reduction is gain, so NAD plus gained electrons in order to form NADH. Note that no ATP is made in this process here, but it's still important to happen because the acetyl-CoA is going to go into the citric acid cycle in order to allow us to make more ATP. And the NADH, with its electrons that it's carrying, it's going to go into the electron transport chain, which is going to help us make more ATP. So this specific set of reactions does not produce ATP, but it produces compounds or molecules that we're going to be able to get ATP from further along in these chemical reactions. So looking at the kind of the product or some of the products of the oxidation of pyruvate, what do you think happens to the carbon dioxide? Hopefully you guessed that we just exhale it for the most part. So it first goes into the bloodstream, and then if we have too much carbon dioxide building up in the bloodstream, we're going to exhale it to get it out of the body. The next question is what happens to the NADH? Remember that NADH is an electron carrier, so it's going to carry electrons to the electron transport chain, which we abbreviate as the ETC. So this is the electron transport chain. The third step in aerobic respiration, so remember the first step was glycolysis, and the second step was the oxidation of pyruvate. So now the third main step is the citric acid cycle, which used to be called the Krebs cycle. The citric acid cycle occurs in the matrix of the mitochondria, so it occurs in the same place as the oxidation of pyruvate. So this is that kind of gooey filled center of the mitochondrion. Now for each glucose molecule that goes through glycolysis, the citric acid cycle is going to occur 2 times. And that's because, remember, we started out with this 6-carbon glucose that then was kind of split in 2 to form 2 3-carbon pyruvates. And then those 3-carbon pyruvates got converted to 2 2-carbon acetyl-CoAs. So taking a look at the overall reactants and products of the citric acid cycle, we're going to start with our 2-carbon acetyl-CoA that goes in. Now that 2-carbon acetyl-CoA goes into the citric acid cycle, and what happens is it ends up releasing that coenzyme A, and one of those carbons breaks off to form carbon dioxide. So we lose another carbon here in the form of carbon dioxide. Now that acetyl-CoA combines with a molecule in order to form another molecule called citrate. And citrate is also known as citric acid, which is how we get the name citric acid cycle, because the first compound produced in the cycle is citric acid. In addition to kind of rearranging and juggling some carbons, we also make some ATP in the mix. So you can see that here that we make some ATP. Now this ATP is made similar to the ATP that was made in glycolysis. So this is also known as substrate level phosphorylation. Additionally, we made some NADH. So remember, NADH is our electron carrier. So we're going to add our little dots to remind ourselves that there are electrons there. So remember that when NAD plus is reduced, reduction is gain. So it's gaining electrons in order to form NADH. Now, in addition to NAD plus, there's another electron carrier called FAD. And it works the same way, where FAD is reduced, which means that it picks up electrons. So it gains electrons. So it's reduced, and reduction is gaining electrons. So once again, we can treat it very similar to the NADH. It's just a molecule, but it's a different molecule than NADH, but the outcome is the same. It picks up some electrons as these chemical reactions are occurring, and it's going to end up carrying those electrons to the electron transport chain. So if we look at the products of the citric acid cycle, let's take a look at what happens to these products. So first of all, CoA, remember this is coenzyme A. And coenzyme A is needed for the oxidation of pyruvate reaction. So it actually gets recycled and goes back to be able to use to oxidize pyruvate again. The carbon dioxide that was produced gets exhaled. So once again, it first goes into the bloodstream, and then as carbon dioxide builds up in the bloodstream, some of it gets exhaled to get it out of the body. The ATP that we made is used by the cell for energy. So we made a little bit of ATP during glycolysis. We made a little bit during the citric acid cycle, and this is ATP that's ready to use by the cell. We also made the NADH and the FADH2. Remember that both of these picked up electrons, and they are known as electron carriers, and they're going to carry or transport those electrons to the electron transport chain. And what's ultimately going to happen is that transferring these electrons to the electron transport chain is ultimately going to allow these molecules to help the cell make a lot more ATP. So now we're moving on to kind of the fourth major step of aerobic respiration. And the fourth step, kind of altogether, we call it oxidative phosphorylation. But remember that oxidative phosphorylation actually kind of occurs in two separate steps. And so the first step is the electron transport chain. And then the second step, I'll go ahead and put it here, is called chemiosmosis. And we'll talk about that a little bit later. So starting by looking at the electron transport chain, we'll take a look at where it's located. So the electron transport chain is actually made up of these transmembrane proteins, which we also learned are called integral proteins. All right, so this chain is just a chain of proteins. So there's multiple proteins that are kind of lined up. And these proteins are embedded in the inner membrane of the mitochondria. And remember, we said that this inner membrane is called the cristae. And the cristae are these folds that go around here. And remember that this inner membrane is folded in order to increase surface area. Because if we increase surface area, we have more room for electron transport chains. And the more room we have for electron transport chains, the more ATP we can make at the same time. So it really helps to make ATP synthesis more efficient. And so these electron transport chains are these kind of proteins that are basically embedded in this kind of inner membrane here. All right, so each set of these would be kind of representing an electron transport chain and an enzyme called ATP synthase that we'll talk about more when we talk about chemiosmosis. So the electron transport chain, what happens is that electrons get passed through a series of these transmembrane proteins and carriers that are embedded in that inner membrane of the mitochondrion. So this picture here is if we were to take a mitochondrion like this and basically zoom into a section of the inner membrane here and look at it in this picture, this is what we would see. So kind of zooming in, we can see that This area here is the matrix. So this is that gooey kind of filled center of the mitochondrion. This area out here is the inner membrane space. So you would have the outer membrane of the mitochondrion kind of like out here. And then this is the inner membrane of the mitochondrion. And you might notice that it looks kind of similar to the cell membrane. And that's because it is very similar in its structure. So as you can see, it is a phospholipid bilayer similar to the cell membrane. So as you can see, we've got these kind of multiple protein carriers and channels that make up this electron transport chain. Now, we're not going to worry too much about the actual names of each of these different proteins. We really care more about what they do. And the main thing that happens is that electrons, as you can see, get passed through these electron kind of protein carriers. Now, where do the electrons come from? You've probably noticed that I'm using the color blue, and throughout the entire lesson, we've been talking about our NADH that I've been writing in blue, and we added that FADH2 when we talked about the citric acid cycle. Now remember, we put those little dots to remind ourselves that there were electrons there. So if we look down here, we've got our NADH and our FADH2. And what happens is that they give up those electrons that they picked up and they pass those electrons through these proteins. All right, you can see that for FADH2 or FADH2 as well, or also. So you'll notice that the NADH gets converted back into NAD+, and the FADH gets converted back to FAD. Now, so far throughout this whole process, we've been talking about how these are aerobic respiration steps, and we've talked about how aerobic respiration requires oxygen. But you may have noticed that we haven't talked about oxygen yet, and that's about to change. So oxygen really comes into play here. So you can see the oxygen here at the end of the electron transport chain. And what happens is that the electrons that come out of the electron transport chain actually bond to oxygen and some hydrogen in order to make water. So basically what happens is you have hydrogen and you have oxygen, and you pick up some electrons and you can make water in the process. So this is why you need oxygen. So this is specifically why you need oxygen in order for aerobic respiration to occur. Now, if you run out of oxygen, so if you stop breathing, what happens is that there's nothing to accept these electrons at the end of the electron transport chain. And if there's nothing to accept it, the electrons can't get passed through the electron transport chain anymore, because it basically gets clogged up with electrons, because those electrons have to go somewhere in the end. And that means that we don't recycle the NADH back into NAD+, and we don't recycle the FADH2 back into FAD. And these two are needed in order to be recycled so that the oxidation of pyruvate and, well, glycolysis, the oxidation of pyruvate, and the citric acid cycle, it needs these to occur. So if we are no longer recycling them, then it means that the cell no longer has the reactants it needs in order to keep going through aerobic respiration. So to summarize, oxygen specifically is needed at the end of the electron transport chain to accept the electrons, and we make some water in the process. And if we don't have oxygen, we can't accept those electrons, and it means that all of the previous reactions shut down. And if those previous reactions shut down, then we can't go through the oxidation of pyruvate, the citric acid cycle, and we can't go into the electron transport chain. All right, so we've talked about these electrons moving through the electron transport chain and ultimately getting picked up by oxygen at the end. But why do we care? The reason why we care has to do with these hydrogen ions here. All right, so these hydrogen ions, so this is just a hydrogen atom, and it's H plus, which means that it's really just a proton. Remember that hydrogen is made up of one proton and one electron, but since it's an ion, since it's H plus, it means that it got rid of its electron. It gave it away. So now, instead of being a proton and an electron, that electron is gone. So we're essentially just talking about protons getting transferred across this inner membrane of the mitochondria. So what happens is that as the electrons are getting moved through these proteins of the electron transport chain, they're providing energy in order to move hydrogen ions from the membrane, from the matrix, which is that inside gooey part of the mitochondrion, and moving it out to the inner membrane space that is between the inner and outer membrane. So the outer membrane would be like up here. And this is the outer membrane of the mitochondria. All right, and so what happens is that we ultimately end up setting up a concentration gradient for hydrogen. So we end up with a whole bunch of hydrogens hanging out in the inner membrane space, and not as many down here in the matrix, because all of the ones in the matrix are getting pumped out to the inner membrane space. So now that we've set up a concentration gradient for hydrogen, so we've got a lot of hydrogen ions in the inner membrane space and not as many in the matrix, we can then use that to basically make ATP. So this is kind of like water behind a dam. So as that hydrogen can move back through another protein, it can actually almost like turn a turbine in order to make ATP. So our kind of the last step of our aerobic respiration, remember that our fourth kind of overall step was oxidative phosphorylation. Oops. And then the first part of that was the electron transport chain. And now the second part of that is going to be our chemiosmosis. So what happens is that these hydrogen ions that are built up in this inner membrane space are going to start going down their concentration gradient. So they're going to go from high concentration to lower concentration. And they're going to do it through a specific protein, which is an enzyme called ATP synthase. And that's what this kind of enzyme is right here, kind of at the end of the electron transport chain. And if we look at the name of this enzyme, it tells us what it does. So ATP synthase. So first of all, ASE tells us that it's an enzyme. Synth should make you think of synthesis, which means to make. And then ATP, remember, is that energy molecule. So this protein is an enzyme that makes ATP. And so what happens is that these hydrogen ions then go down through this ATP synthase, and that includes all of these hydrogen ions that have built up here. They're all going through that ATP synthase. And as they do, that ATP synthase is taking ADP and phosphate and forming a bond between the second and third phosphates, the second phosphate of ADP and then that kind of free phosphate there. And we're ultimately making our ATP energy molecules. So to summarize oxidative phosphorylation, we have electrons that go through the electron transport chain, which ultimately combine with oxygen to form water. As the electrons are getting passed through the electron transport chain, hydrogen ions are getting pumped from the matrix of the mitochondria into the inner membrane space. And this sets up a concentration gradient for hydrogen ions. Those hydrogen ions then diffuse through the ATP synthase enzyme from their high concentration area to the low concentration area. And in the process, it turns that ATP synthase, so it like physically makes it turn in a circle, which basically transfers the energy from that turbine into forming a bond between the phosphate and ATP and a random phosphate that's just kind of hanging out in order to make our ATP. I'm not able to play this video from my in this program here, but if you open up the original Google slide presentation, put it into presentation mode, and then hit play, you can watch this little video that kind of explains what happens during the electron transport chain. And once again, I cannot play this video in this current program that I'm using, but if you go to the original Google slideshow, you can put it into presentation mode and then hit the play button in order to watch the animation, which kind of helps to explain what happens during chemiosmosis. So let's take a look at how much ATP we made from cellular respiration. And remember that with cellular respiration, we're more specifically referring to aerobic respiration. So this was with oxygen, right? So we had oxygen involved, and this was going through those kind of those 4 stages of glycolysis, the oxidation of pyruvate, the citric acid cycle, and oxidative phosphorylation. So for each glucose molecule, so per glucose, here's what we get with regards to ATP production. So during glycolysis, we made 2 ATP. We actually made 4, but remember we had to invest 2, so we end up with a profit of 2 ATP. During the citric acid cycle, we made an additional 2 ATP. Now, the real kind of payoff actually happens during that oxidative phosphorylation, where remember the NADH with the electrons and the FADH2 with the electrons go through electron transport chain and chemiosmosis. Remember, during chemiosmosis, we had that enzyme called ATP synthase. So the end result from all of these reactions kind of put together, we end up with about 36 to 38 ATP molecules made per glucose molecule. So one glucose molecule, which is C6H12O6, provides us with a total of about 36 to 38 ATP. And that includes 4 ATP from the substrate level phosphorylation, and then about 32 to 34 ATP from our oxidative phosphorylation. So let's go over the kind of the summary of the reactions of aerobic respiration again, so that we can maybe more easily follow what's happening with the carbons, the ATP, and the NADH and FADH2. So first, let's start with glycolysis. So remember, in glycolysis, we start out with glucose, and glucose is a 6-carbon compound, and that glucose gets converted to pyruvate. Pyruvate is a 3-carbon compound, and we actually make 2 of those because of the glucose getting essentially chopped in half. Now that pyruvate then goes through the oxidation of pyruvate in order to form acetyl-CoA. And so the acetyl-CoA has coenzyme A coming in to form the acetyl-CoA. And we also have some carbon dioxide leaving. Now that acetyl-CoA goes into the citric acid cycle. And during the citric acid cycle, we do produce some carbon dioxide. Now remember that during these processes, we're also making some ATP. So during glycolysis, we actually have to put 2 ATP into the chemical reaction to get it started. And those 2 ATP get broken down into 2 ADP plus P. But we also make 4 ATP. So we have 4 of the ADP plus phosphate going in, and we end up with 4 ATP coming out. So altogether, we end up with plus 2 ATP during glycolysis. Now, during the oxidation of pyruvate, we do not make any ATP, but we do make some during the citric acid cycle. So we end up with 2 ADP plus phosphate going into the citric acid cycle, and we produce 2 ATP coming out. Now, in addition to basically rearranging or juggling these carbons and making ATP, we're also doing the redox reactions with our electron carriers. So during glycolysis, we have NADH go into the reactions where it picks up some electrons in order to form NADH. So NAD plus picks up electrons to form NADH. That also happens during the oxidation of pyruvate, where some NAD plus goes into the reactions where they pick up some electrons to form NADH. During the citric acid cycle, this happens with NAD plus, again, picking up electrons to form NADH. It also happens with another electron carrier called FAD, which picks up electrons to form FADH2. Now remember that the electrons picked up by NAD plus and FAD to form NADH and FADH2, these electrons go to the electron transport chain, So these electrons all go to the electron transport chain, which allows for oxidative phosphorylation to happen through chemiosmosis. So with these electrons going through the electron transport chain, we end up making an additional roughly 32 to 34 ATP. So once again, the conversion of glucose to pyruvate is known as glycolysis. The conversion of pyruvate to acetyl-CoA is known as the oxidation of pyruvate. The citric acid cycle is what's happening here. And then the electron transport chain is the first part of oxidative phosphorylation. Which includes the events of the electron transport chain followed by chemiosmosis. So once again, glycolysis is first, followed by the oxidation of pyruvate, then the citric acid cycle, and then oxidative phosphorylation. So now that we're done talking about aerobic respiration, we need to circle back around and take a look at what happens during anaerobic respiration. So remember that our cellular respiration has 2 pathways. Remember, we have the aerobic, which we just talked about, and the anaerobic. And the main difference between the two is whether there is oxygen or not required for the reactions to happen. Now, remember that the first step of both aerobic and anaerobic respiration is glycolysis. But what happens to the end product of glycolysis is going to differ. So remember that during glycolysis, you have a glucose, which is a 6-carbon compound that gets broken down into 2 pyruvates, which are 3-carbon compounds. Now, if oxygen is present, that pyruvate goes through those remaining steps of aerobic respiration. But now we're going to circle back around and take a look at what happens when oxygen is not present. So if we can't do aerobic respiration, what's going to happen to that pyruvate that we produce during glycolysis? Well, the answer is it's going to go through another series of reactions. So instead of going through the oxidation of pyruvate and the citric acid cycle and oxidative phosphorylation, instead it's going to go through something called fermentation. Now, it's important to note that the fermentation reactions don't actually produce ATP. So if fermentation doesn't produce ATP, why do we care about it? Why do we care about these fermentation reactions if we don't get any ATP out of it? Because the whole purpose of respiration is to make ATP. So this picture here, if we were actually in a classroom, I would have you sit there with your hands held out to the side. And you can still do this exercise at home if you want to. You'll notice that as you hold your arms out to the side, your arms start to get tired, and it makes it harder to keep them up, and they might even start to get kind of sore and weak, and over time you're going to hit a point where you are not going to be able to hold your arms up. And that's because eventually things like your muscle cells might start doing anaerobic respiration instead of aerobic respiration, and that can actually lead to muscle fatigue. So there are a couple of different fermentation pathways. There's actually lactic acid fermentation, and there's alcohol fermentation. And these different types of fermentation happen in different organisms. But let's take a look at some of the products or what comes out of these reactions before we talk about which organisms do which. But first of all, we mentioned about how muscle fatigue happens because of fermentation, and this happens because of lactic acid fermentation. So human, really any mammal, the muscle tissue, if there's not enough oxygen present to go through aerobic respiration, instead those cells are going to go through anaerobic respiration. And one of the end results is lactic acid, which leads to muscle fatigue. Fermentation also produces a lot of the stuff that we enjoy eating. So for example, yogurt is actually lactic acid fermentation in bacteria. Alcohol is produced from alcohol fermentation in yeast. Bread is also from yeast. Sorry, I did those in reverse order, but now, in addition to that, there are other things like, so like we talked about beer here, we talked about our yogurt, we talked about bread, also stuff like kombucha. So kombucha is actually made using bacteria and yeast that go through these fermentation reactions. Things like sauerkraut and kimchi, these are all things that are produced using these fermentation reactions. So as we mentioned before, fermentation itself does not produce ATP, but remember that glycolysis happens before fermentation. So our anaerobic respiration, we have glycolysis happening first, and glycolysis, remember, makes 2 ATP. And then our second step is fermentation. Now fermentation, like we said, it does not make any ATP. So 0 ATP, which we would think would be a bad thing, right? Because we want to make ATP from these glucose molecules going through glycolysis. But the reason why fermentation is still important is because it actually helps to recycle the materials needed for glycolysis to happen again and again. So if we look here, we've got our glucose going through glycolysis in order to create our pyruvate. And we make some ATP in the process, which remember ATP is energy. We're also making some NADH in the process. So remember NAD plus picks up some electrons from those chemical reactions to form NADH. Now NAD plus is a reactant of glycolysis. So we have to have NAD plus in order for glycolysis to occur. So what happens is these fermentation reactions play an important role in that they take the NADH and they actually remove those electrons in order to convert it back into the NAD plus so that glycolysis can happen again. So fermentation itself does not produce ATP, but fermentation takes NADH and oxidizes it back into NAD plus. Remember that oxidation is loss. So NADH loses some electrons in the process, and NAD plus is made as a byproduct. So it basically recycles. So fermentation recycles NADH back into NAD plus, and then that NAD plus can go into another glycolysis reaction. So fermentation is really important because in order for us to make the ATP in glycolysis, we have to recycle that NADH so that glycolysis can happen again. So to summarize our anaerobic respiration, remember that we're starting with an organic compound. So we'll start with our idea of just glucose, which remember is a 6-carbon compound. During glycolysis, that glucose gets converted to pyruvate, which is a 3-carbon compound, and we make 2 of them because glucose is essentially chopped in half. During fermentation, that pyruvate gets converted to an end product, which may be either lactic acid in some organisms, or it might make ethanol and carbon dioxide. During the glycolysis reactions, some ATP is made. So just like we talked about with aerobic respiration, we have to put 2 ATP into the reactions in order to get them started. But we ultimately get 4 ATP produced by those reactions. So we end up with a net of plus 2 ATP made during glycolysis. Now, we also take some NAD plus and we pick up some electrons in order to form NADH. Now, the fermentation reactions do not produce ATP, but what they do is they recycle NADH back into NAD+. And NAD+ is a necessary reactant for glycolysis. So if this doesn't happen, then eventually we're going to run out of NAD+, and glycolysis is not going to be able to happen. So as a reminder, this top arrow up here is glycolysis. This bottom arrow down here is fermentation. And the main goal or reason why fermentation is important is to recycle that NAD+ so that glycolysis can happen again so that we can make more ATP from additional glucose molecules. A couple more facts about fermentation. So it is an anaerobic process, so oxygen is not required for the reactions to occur. Fermentation occurs in the cytoplasm, so we don't need a mitochondrion in order for fermentation to occur. And in fact, things like bacteria don't have mitochondria, but bacteria can do these fermentation reactions. And the end products depend on which organism is going through these fermentation reactions. So some bacteria and the muscle tissue of mammals produces lactic acid. Now, in humans and other mammals, that lactic acid is likely what produces muscle fatigue, which is what makes your muscles weak after a lot of use. In bacteria, that lactic acid is what gives things like cheese and yogurt its kind of tanginess to it. So you can actually make yogurt by taking milk and adding a culture of the right type of bacteria, and the bacteria go through that fermentation reaction to form the lactic acid, which is how you make yogurt. Yeast is a eukaryotic organism. It's a type of fungus. And yeast goes through alcohol fermentation. So it produces ethanol, which is the alcohol found in the alcohols that you drink. And it also makes some carbon dioxide in the process. And that carbon dioxide is actually what makes bread rise as well. So if you're making bread from scratch, you take something like flour and maybe a little bit of sugar, mix it with water and yeast, and the dough rises because carbon dioxide is being produced by the yeast, and that gas gets kind of trapped inside the dough. So now if we compare the amount of ATP that's made, if we look at aerobic respiration versus anaerobic respiration, we can take a look at another main difference between these two types. So remember that aerobic respiration requires oxygen, whereas anaerobic respiration does not. That's one key difference there. The other key difference is that for each glucose molecule that goes through aerobic respiration, we can produce about 34 to 36, somewhere around there, ATP molecules. With anaerobic respiration, for each glucose molecule that goes into the reactions, we only make 2 ATP. So it really comes down to return on investment. So you can imagine that as an organism, it takes energy and time to go find those organic nutrients such as glucose. So if you had to invest time and energy into getting these organic molecules into your body, do you want a return on investment where you get 34 ATP per glucose molecule, or only 2? All right, so as humans, we actually really rely on this aerobic respiration. Our bodies can do fermentation, or at least some cells in our body can do fermentation, but it's not enough to keep us alive. So in order for survival, humans have to breathe so that we can take in oxygen, so that we can have a much higher efficiency with regards to producing ATP per glucose molecule. Now, not all organisms rely on aerobic respiration. So for example, there are some bacteria that only do anaerobic respiration, and they can survive on that just fine. But humans, that's not the case. Humans have to do at least the majority of the respiration has to be aerobic respiration. So far, as we've talked about these reactions for aerobic and anaerobic respiration, we've talked about starting with glucose, going through the process of glycolysis in order to form pyruvate. So that was the first step of both aerobic and anaerobic respiration. But remember that glucose is not the only molecule that we can get ATP from. We use glucose as an example as we go through these reactions because it goes through every single step of all of these reactions. But in reality, we could basically replace the word glucose with the word organic compound. All right, and it's not just any organic compound. More specifically, we're talking about our kind of carbs in general, our lipids, and proteins. And it really all depends on which enzymes are present in order to break down the specific organic compounds. So we can't get energy from every single organic compound. We can't get it from every single carbohydrate or every single lipid or every single protein. But depending on which enzymes we have present, it's not just glucose. We can also get energy, for example, from fructose or sucrose, or fats and oils, or eating proteins in the foods that we eat. So let's first take a look at some of the other carbohydrates that we can get ATP from. So other sugars that we ingest are typically first converted into glycogen, and glycogen is a polysaccharide. Remember, poly means many, and so glycogen is basically a whole bunch of sugars bonded together. And this is actually how our body stores carbohydrates, and this is typically being stored in the liver. So first, things like fructose and sucrose might get converted to glycogen, and then glycogen gets broken down into its individual units, which are glucose molecules. And so each one of these glucose molecules can then go through glycolysis to form pyruvate, and then it can go through the remaining reactions of cellular respiration. Proteins have to be catabolized or broken down into their amino acids first before they can be used to make energy, or ATP. Now, there are 20 different types of amino acids, and so the different amino acids actually go into the different reactions of cellular respiration at different areas. Now, this image down here, you do not have to memorize this image. I'm just using this to show you kind of what's happening with these amino acids. So this image is showing you up here, we're starting with pyruvate. So we're looking at the oxidation of pyruvate here, which forms our acetyl-CoA. And then acetyl-CoA is going into our citric acid cycle. Now, all of these kind of compounds in these boxes, these are showing us the amino acids. And so depending on what the amino acids are that made up that protein, they're going to enter into the reactions in different areas. So for example, alanine is an amino acid, and alanine kind of starts at the beginning of the oxidation of pyruvate and then goes through the remaining steps of cellular respiration. Something like arginine down here first gets converted to glutamate, which then goes into one of the intermediate reactions of the citric acid cycle. So like I said, you don't have to memorize this. Just know that proteins are used for energy by first getting broken down into their amino acids, and then the amino acids go into the different reactions of cellular respiration, depending on what kind of amino acid they are. And last but not least, we're talking about our lipids. So more specific, our dietary lipids are called triglycerides. So these are the dietary lipids. These are the ones that we ingest from our food. And these triglycerides are things like fats and oils. So when you're eating olive oil or butter or lard, you're eating triglycerides. Now, triglycerides are made up of a glycerol molecule bound to 3 fatty acid molecules. So we kind of draw them with like a glycerol, and then they've got these like 3 fatty acids that kind of stick off of them. So the first thing that happens is that the triglyceride gets broken down into the glycerol and the 3 fatty acids. Glycerol then gets converted to something called G3P, which is glycerol 3-phosphate, which, as you can see over here, is an intermediate of glycolysis. So it kind of skips the first couple of reactions of glycolysis, and it just starts from this point on. Fatty acids get converted to acetyl-CoA that goes into the citric acid cycle. So once again, you don't need to know the nitty-gritty specifics of this, other than just know that the triglycerides get catabolized first into their smaller components, and then those smaller components can go into the reactions of cellular respiration, but they don't start at the beginning. They start somewhere further along in the process, but we can still get ATP from them. Now, we talked about the first law of thermodynamics, which states that Energy is neither created nor destroyed, but it can be passed around from organism to organism. And as we went through the reactions of cellular respiration, we talked about energy in the form of electrons getting passed between glucose and other carbon molecules and then getting transferred to NAD plus to make NADH, and then also getting transferred to ATP or ADP in order to make ATP. And so we talked about how those chemical reactions are basically just moving energy around into different molecules. Now, the second law of thermodynamics is also really important as we talk about cellular respiration. And the second law of thermodynamics essentially states that these energy conversions are not 100% efficient, meaning that if you have, let's say, 10 units of energy and you're trying to transfer those 10 units of energy from glucose to, let's say, NAD plus, not all 10 of those units are going to make the transfer successfully. So some of that energy is actually going to be lost in the process of being converted from one form to another. And in most cases, this energy is lost in the form of heat. So for example, we used the example before about putting gasoline into a car and how gasoline is basically just chemical energy, and the car is able to break those bonds in the energy to convert the potential energy to the kinetic energy of the car moving. Now, you've probably gotten out of a car after driving it for a while, and if you touch the hood, you'll notice that it's really hot. That's because as the potential energy in the gasoline gets converted to the kinetic energy of driving the car, some of the energy gets lost in the form of heat in that process. And you want to know what really stinks is about 90% of it gets lost as heat. So out of all that gasoline that you pay for and put into your car, only about 10% of it is successfully converted into a form that moves your car. The rest of it is lost as a waste product known as heat. Now, that sucks when it comes to your car, right? Because it seems like you're wasting money. But in humans, this is actually a really good thing. And that's because humans and other mammals are warm-blooded, which means that we maintain a relatively stable internal body temperature, even if it gets really cold outside. And you've probably noticed that or experienced that when you get really cold, you start to shiver. And what shivering is, it's just uncontrolled or involuntary skeletal muscle contractions. So what's happening is that your skeletal muscle is taking energy stored in the form of these organic compounds, converting that energy to ATP, which then gets converted to the contraction or the movement of the muscle. But in this case, the movement is actually kind of like the side product or the side effect, because what the shivering is ultimately trying to produce is that heat in order to warm you up. All right, so the second law of thermodynamics when it comes to living systems like humans is actually really beneficial, because that heat can help to warm us up when we get too cold, which helps us to maintain our homeostatic body temperature.